Hi everyone, welcome back. Today's goal is to discuss Chapter 4, which is entitled The Energy of Life. Chapter 4 has quite a few concepts, so it has been divided into two parts. Our first part will focus on energy, the laws of thermodynamics, and a discussion to figure out what the difference is between an endergonic and an exergonic reaction. Now let's just go ahead and start off with a basic definition, which is what exactly do we mean when we use the term energy? Well, from a biology sense, it's going to be the cell's ability to do work. And when we take a look at energy, we can go ahead and divide it into two broad categories. You have energy that you are currently using, and you have energy that you are storing or saving for a later time. So on your slide, you're going to notice that there is a mention of what they call kinetic energy, and there is potential energy. Kinetic energy is often called energy of motion. It is energy that is actively being used to complete work. So for instance, if you're holding onto a pen right now and scribbling down some notes, that is because of kinetic energy. Perhaps you're eating a snack, so the fact that you are chewing and you're holding on to your utensils, that is all because of kinetic energy. If you are someone who drives a car, anytime you get in the car and you start driving, the movement of the car is an example of kinetic energy. Now, potential energy, as the name indicates, has stored energy which will be available to do work at a later time. For many of us, the best example of potential energy can be found in the foods that we consume, because the foods, the carbohydrates, the proteins, the lipids, they are full of chemical connections and chemical bonds, which are holding on to energy that we are trying to pull apart and retrieve through our digestive system. Another example, if you want to think about it for potential energy, going back to the car, is if you drive, then, when you get to your destination, you'll park your car, you'll get out the car, and you go about your day. As the car is sitting there in its parking space or in your garage, that car is filled with potential energy. And what will end up happening is that the minute you get in the car and you turn on the ignition, the potential energy is then converted into kinetic energy. Even if we take a look at the illustration at the bottom of our slide, we see that we have a bicyclist. Now, the actual motion of pedaling the bike, that would be an example of kinetic energy. And his body will also have potential energy in the form of perhaps a protein bar that he consumed before he started pedaling. But also what you might notice is when they get on top of the hill, as they start sliding downwards, we can utilize the energy from gravity as the source of getting the work done. So energy truly is all around us. And in order to better understand us, I wanted to introduce the first two laws of thermodynamics. And I think if you kind of see what they aim about, it's going to make a little bit more sense as to how kinetic and potential energy are linked together. So let's start off with the first law of thermodynamics. This one basically says that energy cannot be created or destroyed. It simply is converted from one form to the next. So you don't create nor destroy energy, you simply convert it from one form to the next. And the total amount of energy in the universe is constant. And part of that we can explain by taking a look at a food web. So here we have an example where we have the sun providing its sunlight, which is an example of kinetic energy. And organisms such as this tree and any other autotrophs are able to take this energy and then convert it through the use of photosynthesis into energy that the plant can utilize. Example, glucose. Now, the glucose is then a form of potential energy for the plant to use at a later time span to maintain or to grow. Or better yet, what if we then introduce another animal into the system? In this case, we have a deer. Now remember, deers are heterotroph, so in order for them to extract energy, they're going to have to consume another organism, whether it's an autotroph or a heterotroph. So in this case, this deer is chewing on the leaf, and as the deer is doing that, what we see happening is that the energy is being transferred from the plant over to the deer, And what we're seeing here is the example of the first law, which is that no energy was created, no energy was destroyed. It was simply converted from one form to the next. So in this case, it would be the interplay of the sunlight to the autotropes through the heterotropes. And even within the cells, we see that the cells are able to go ahead and do this thing called cellular respiration, which basically means that they're going to be able to extract the energy that's in the food, and as they're doing hydrolysis, they're presenting it over to the mitochondria so that the mitochondria can go ahead and convert that energy into another form, more specifically ATP molecules. So the first rule of, or I should say the first law of thermodynamics is that you do not create or destroy energy. You simply convert it from one form to the next. The second law is also referenced on this item or in this image. And you can see right here that every time there is a movement from one organism to the next or one process to the next, there is some heat that's being lost. See the little red arrows? That leads us to our second law. And it says that energy transformations are inefficient. It is never 100% transfer of energy from point A to point B. Instead, every reaction that occurs will lose some of the energy in the form of heat. Now, heat is very random, it's very disorganized, so we can't utilize that as an effective energy source. Instead, what we can see happening is because of the high amount of disorder, we're going to generate what's called entropy. Entropy is the measure of randomness. We cannot reverse this heat loss, and the more the energy is passed on from one organism to the next, the higher levels of entropy will be accumulated. So the first law is that you cannot create or destroy energy. You simply convert it from one form to the next. So for instance, from the sunlight to an autotrope to a heterotrope. And the second law says that these transformations, these movements from point A to point B, are never 100% effective because along the way you lose heat, which is classified as being very random and disordered. So you could say that you generate lots of entropy. Now, for the cell itself, what we see happening is that energy has many different uses. It's going to allow us to build, it's going to allow us to maintain, it's going to allow us to grow, to produce, lots and lots of functions. And part of that will then be encompassations under the term metabolism. The metabolism takes a look at all the different chemical reactions that are related to energy, whether that energy is being absorbed or released. So within our metabolism umbrella, we have two different functions. We have what we call an endergonic reaction, and we have an exergonic reaction. Endergonic reactions are going to focus on how we use an input of energy to build or to generate new molecules or reactions. An exergonic reaction is going to do the opposite. Exergonic reactions are going to focus on how we release energy, and the release of that energy is usually due to the fact that we are doing a breakdown or a hydrolysis of complex molecules. And by breaking down these molecules, we break down the chemical bonds, thereby releasing the energy that was contained inside of them. So endergonic focuses on building, and that will require an input of energy, whereas exergonic will focus on destroying or breaking down, example, hydrolysis. And as it's breaking down, it's releasing the energy that was stored within the chemical bonds of the complex molecule. You can think about it also in the sense of a construction site. So here on the illustration from the book, you can see that on the top part right here, they're actively building. And in order to build, that would mean that you're putting the smaller parts together to form something larger. So we're thinking hierarchical pattern, we're thinking emergent properties, but we're also thinking something along the way of our body when it, for instance, wants to build muscle, it needs to find amino acids and link it together. Or in our example from our previous chapter, when we were looking at milk production, that entitled us to collect many different parts so that we can merge it together. anytime you're actively building, what we see happening is that will be classified as an endergonic reaction because you're going from simple to complex, and that will require energy to be put into the system. So you require energy to get it done. On the other hand, if we go back to our picture, we can see that the bottom one for the construction is a demolition site, and we see how the building is collapsing upon itself. This is the same thing that we happen whenever we have a large molecule that we are breaking down in our body. So perhaps our digestive system would be a good example in this case. Anything that you had for your last meal is currently being hydrolyzed or broken down by your digestive system. This would be an example of an exergonic reaction because your goal is to take a larger molecule and break chemical bonds so that it breaks down in smaller pieces. And as the smaller pieces are formed, the energy is released that was contained within. So an exergonic reaction will release energy into the system, and an endergonic reaction requires energy to put into the system. Now, turns out both of them are dependent on each other because the energy that you release from an exergonic reaction, I wrote down, well, what happens to it? Well, this energy will go ahead and be directly funneled into the endergonic reaction. So one releases it and then one uses it. It is a consistent flow of energy as you build and you destroy. And even on the electron level, we see that this is happening. And what we tend to do is we use the terminology oxidation reduction. When you see that term, I want you to think about two things. First, I want you to think about the fact that we're talking about electrons. And the second thing I want you to think about is that for these electrons, part of it will be lost. The part that you lose, or when you lose those electrons, that is called oxidation because you are breaking apart the electrons. It is getting smaller, so it's an example of an exergonic reaction. And those electrons that are lost will need to find a new home or a new element, and that would then be an example of a reduction. A reduction is the aspect that receives the electrons, so it's building upon it, and it's an example of an endergonic reaction. Now, oxidation reductions occur simultaneously, so you will always have what we like to call an electron donor who gives it away, and you will have an electron receiver, so that the oxidation and the reduction are occurring together. Please keep in mind that this is a reference for the movement of your electrons. Here is an illustration from the book, and as you can see, you have two lovely ladies who are doing a gift exchange. The first lady in blue with her hat, she is going to represent our electron donor, and she will be giving away the electrons as symbolized by the gift right here. And as the electrons are released, we're going to say that's an oxidation process that's occurring. The molecule that stays behind has been oxidized. Now these electrons need to find a new home, so the gift will be moved over to the electron acceptor, who will now house these two electrons now. So we can say that this is the reduction, and this would be a reduced molecule. So oxidation reduction, it's another look at exergonic and endergonic, but only exclusively on the electron level. And the reason we mentioned this is because we just finished our chapter where we were talking about the cell and the cell membrane. Remember the little phospholipid bilayer with the hydrophilic heads and the hydrophobic tails? So here's your little plasma membrane, and embedded in the plasma membranes, you have different proteins that are able to complete what we call an electron transport chain, which is that they can find electron donors, and the molecules will release the electrons, and the electron will funnel from one protein to the next, releasing energy along the way. It's a fantastic way for the cells to obtain little bursts of energies. And as the energy becomes depleted or lower, then the electron will go ahead and find an acceptor molecule so it can go ahead and be reduced. So you have your oxidation, the release, and you have your reduction, the acceptance of a new molecule. Very effective way for your cell membrane to obtain some energy. Now, of course, we can't talk about energy without mentioning the mitochondria. The mitochondria is the powerhouse of the cell, and within that, we see another example of how energy will be converted into another form. What do I mean with that? Well, anytime you sit down and you consume a meal, part of why we are eating is because we want our body to break down the foods, the carbs, the proteins, and the lipids. We want them to do hydrolysis, which is an example of an exergonic reaction, so that we can release the energy that's being held within the bonds of our food molecules. That energy will eventually be presented to the inside of the cell, and the cell will automatically funnel that energy over to the mitochondria. The mitochondria will convert that energy into ATP molecule. ATP stands for adenosine triphosphate. And the magic of this molecule is that it's able to store the energy between the three phosphate molecules right here. So by linking together three of these phosphates, the tri stands for three, it is able to store the energy that was presented to the cell and brought over to the mitochondria. So an ATP molecule is often indicated by a little sunburst. And that is your hint to realize that it is a charged molecule, a molecule full of energy. And if the cell then requires energy, it will go over to the mitochondria and it will say, hey, can I have some energy? The mitochondria will give it ATP. And the way that the cell will be able to harvest the energy in the ATP molecule is by breaking one of the phosphates off. And by breaking one of the phosphates off, it's almost like releasing the cap of a container. It will release the energy that's being held within the ATP molecule. So in this little formula right here that you see at the bottom of your slide, you see that you can take your ATP molecule, remember that means 3-phosphate, and when you break it apart, so you do hydrolysis, when you break it apart and you tear one of the phosphates off, what's going to happen is, here's my phosphate being torn up, is that my ATP molecule is going to be converted into an ADP molecule. D stands for di, which means it now only has 2. The ADP molecule, notice how it doesn't have a sunburst around it because it's not charged. And that is because when you remove that one phosphate and left the ADP molecule, you also released the energy that was being housed inside of the ATP molecule. All of the cells that we have in our body depend on this consistent formation and breaking down of an ATP molecule. Please keep in mind that ATP is the charged molecule. It has energy, and when you need energy, your cell will ask for ATP and it will break it open. It will do hydrolysis on it. And when it breaks it open, you will be left with a phosphate, ADP, and the energy that you are looking for. So we can take that energy and then complete our different endergonic reactions that require an input of energy to be done. And your body literally produces billions of ATP molecules consistently. It's just this continuous play of building and using, building and using. And when you look at an illustration like the one you have right here, what you see is the connection between how we talk about endergonic, exergonic, ATP, ADP. So here, for instance, we see if we start on our left-hand side, we see that every time we consume a meal, in this case, we're back to our bicyclist. So he had a little energy bar. Your body, your digestive system knows that this energy bar is full of energy. So it will go ahead and do an exergonic reaction. It will break the bar down from polymer to monomer. And as it breaks down, it hydrolyzes the food. We see that the energy that was being held between the bonds is released and it's converted by the cells into ATP. Thank you, mitochondria. As soon as the ATP molecule is produced, we can go ahead and just use hydrolysis again to break it down into ADP and a phosphate and release the energy that it was housing. This energy can then be plugged into an into an endergonic reaction so that we can build. So like for instance, we can build muscle tone, we can build stamina, as we can build endurance as the bicyclist continues his journey of pedaling the bike. So exergonic releases energy, your body is able to produce ATP molecules, and as soon as you have the ATP molecules, we can destroy it again, leaving behind the phosphate and ADP and releasing the energy that it houses. It's just this continuous process. And please do keep in mind that everywhere you see the mention of your H2O, that is just hinting at the fact that you're doing your dehydration and hydrolysis synthesis, depending, of course, on if you are building your ATP molecule or if you are destroying it. Your body is not able to store ATP molecules. So part of your homeostasis is to figure out how much you need on a continuous basis so we don't stockpile because the molecule is simply too unstable between those three phosphates to keep it for long-term storage. Instead, what your body will do is it will store energy-rich molecules. So for many of us, that, for instance, would be something like fat. Anytime you consume too many calories and your body can't utilize the energy right off the bat, it will store some of it in the form of fat. This fat can then be used later on. So this is one of the reasons why if you're looking into losing some weight, a reduction in calories, so going on a diet, as well as exercise is an effective way because when you exercise, you use more energy. And if you're not getting that energy from the food, then your body has to go over to the adipose tissue, your fat cells, and harvest the energy that's stored in there. You don't automatically make fats if you eat a little bit too much because it is a multi-step process. Most of the time, your body will store some of the extra energy as glycogen, which is a temporary storage unit, and it keeps us kind of filled between meals. Also, plants are a good example of utilizing starch as an energy-rich molecule that it would like to store for a later day or time. Now, because your energy use is so consistent throughout the body, we can kind of use a little bit of help to try to save on some of this energy, and that is then where our next topic is going to lead to. On this part of the chapter, we're going to take a look at what we call enzymes. Enzymes are classified as biocatalysts, and a catalyst means that it speeds up. In this case, the enzyme is going to speed up chemical reactions, and it's going to do so by lowering the total amount of energy that is required for activation. By lowering the energy that's required for activation, it will allow the reaction to get started quicker and to get to its final goal at a faster rate. The nice thing about an enzyme is that the majority of them are protein-based. You do have a few that are RNA-based, but the majority of them are protein-based. And an enzyme has a few requirements that we need to discuss before we can successfully use it. But remember, its ultimate goal is to allow for the reaction to occur at a faster rate. So here on my little diagram, I have two scenarios. I have activation enzyme without an enzyme. So as you can see, quite a high peak that we have to go through. And the purple one is significantly shorter because that is going to have the use of an enzyme, which will automatically lower the activation energy that the cell has to put into it for the reaction to occur. Now, when you use an enzyme, you save energy, the reaction occurs quicker, but you still get the same products, which is fantastic. And even better, you get to recycle the enzyme. You can use it over and over again. The part of the enzyme where all the magic happens or where all this activation energy is reduced, it's called the active site. And it turns out that enzymes have specificity, or what we call a fit-induced model. Enzymes are specific as to what reaction they can activate. So we like to call that a substrate. A substrate is what will activate the enzyme. And in order to activate the enzyme, it has to fit within its active site. So I always like to think of it as a lock and key analogy. So the proper substrate will properly fit in the active site and it will unlock the enzyme, allowing it to lower the activation energy and keep working until the products are ready to be released. Now, imagine that you then have enzymes for a wide variety of substrates. So, for instance, in our digestive system, we have enzymes that are specific for carbohydrates, we have enzymes that are specific for proteins, we have enzymes that are specific for lipids, and this is all based on the fact that the substrate has to fit on the active site. If it doesn't fit, the enzyme will not get activated and there will not be an increase of production of product being produced at a shorter time span. So here, once again, you have another illustration of how the active site of an enzyme will interact with the substrate, and it's only when the substrate fits that you can go ahead and activate the pathway and you can get to your products faster. A lot of the times, the name of the protein will indicate that it's an enzyme because they'll utilize the same three letters, ASE. So for instance, lipase, amylase, lactase, that ASE hint is just for us to kind of get a quick reminder that we're dealing with an enzyme. Now, your enzymes, though, also need to have a way, in essence, to be controlled as to when we want them to be active or not, because obviously we don't want to waste any energy on maintenance on something that we don't actively need at the moment. So what we see happening is that cells are able to control their reaction rates by using feedback mechanisms. And you have what we call a negative feedback, which is the most common, and you also have a positive feedback. A negative feedback basically means that we're only going to use the enzymes when we need to. And as soon as we get to our results and the product, we're going to shut the enzymes back off, excuse me. This is a perfect example of a supply and demand scenario. So what I'm telling you is that in a negative feedback, the enzyme will receive a stimulus, it will turn on, and the minute the enzyme is done with its work and has produced a product, the enzyme will shut down. So when you take a look at the illustration that's at the bottom of the slide, you're going to notice that you have 4 enzymes that have been drawn out for us, and they are part of what we call a cascading pathway, where one enzyme leads to the activation of the next enzyme. So as you can see right here, when receiving the stimulus, enzyme number 1 will bind and interact with its proper substrate and produce the product that will go ahead and bind with enzyme number 2's active site. And you just keep going. The product of one leads to the activation of the other. And by the time you get to enzyme 4, you get to your final product. What the negative feedback mechanism tells us is that the minute you get your product, you are done. You have met your demand. Well, then we're going to send the signal and we're going to prevent any further workings of enzyme 1, 2, 3, or 4. And we won't turn these enzymes on again until we receive another stimulus that it's time to produce more product. I want you to think about an example that we use. For instance, let me do an example that I use in my house all the time. So I have my air conditioning running. I like it to be relatively comfortable in my house. So I like to have my air conditioning at about 70 degrees Fahrenheit when I'm walking around. Now, when I leave the house, though, it doesn't need to be that cold. So I'll go over to the thermostat and I'll set it, maybe at like 75, 77. I'll go about my day. And then when I get back home later on the night, the first thing I'm going to notice is that it's warmer in the house than I would like it to be. So I'm going to go over to my thermostat and I'm going to put it and send a stimulus that I would like it to be 70 degrees in the house. Please keep in mind this is Fahrenheit. What's going to end up happening is that that stimulus through the thermostat is going to activate the AC unit, and the AC unit is going to start pumping air into the house. And it's going to keep working until it produces enough cold air for the house to go down to 70 degrees Fahrenheit. When that happens, the AC unit shuts down. And it shuts down because we don't want it to be colder, and it also shuts down because we don't want to waste any additional energy. That is a perfect example of a negative feedback system. You turn on when needed, and then the second you have your product, you turn off. Now, how do we turn off? Well, for the enzyme, it turns out that it likes to use competitive and non-competitive inhibition. So take a look at your illustration at the bottom, and you can see the first one is one of normal binding. You have your beautiful enzyme, it has its specific active site, and the substrate will perfectly bind to it, and the enzyme will turn on and do what it needs to do. If we don't need the enzyme, then what we're going to do is we're going to inhibit the substrate from interacting with the active site. And we can do that two ways. The first way is to do competitive inhibition. And competitive inhibition means that I'm going to be competing with the active site, and I'm going to put an inhibitor. So this could just be a random protein or just any structure that I'm just going to put in the way of the active site. So when the substrate comes, there's nowhere for it to bind because something is blocking or competing for its active site. So this would be competitive inhibition when you have an inhibitor binding to the active site. So there's nowhere for the substrate to bind. The other option is called non-competitive. And in non-competitive inhibition, you take the inhibitor as well, but now you bind it somewhere else besides the active site. And when you bind it somewhere else, what we see happening is that the shape of the active site will indirectly change. And once again, the substrate cannot bind to this new shape. So you also shut down the enzyme. So notice how in both cases, you basically interact or alter the active site, but the difference is a competitive inhibition, you are directly blocking the active site. In non-competitive inhibition, you are binding an inhibitor elsewhere and thereby indirectly changing the active site. In both scenarios, the substrate is not able to bind, so the enzyme does not turn on. Now, I told you there was a negative feedback and then there's a positive feedback. So if negative feedback tells us we are going to receive a stimulus, we are going to do the work. Once we receive our product, we shut down. Positive feedback is going to do the opposite. Positive feedback says, yes, I will receive a stimulus. Yes, I will produce product. But when the product is produced, instead of shutting down, I am going to amplify. So I'm going to increase the work and I'm going to produce even more product. This is an amplification pathway. This requires lots and lots of energy because you're continuously working on the process. So the body tends to use positive feedback for life-altering or perhaps life-saving situations. So for instance, blood clotting would be a good example of a positive feedback system. In your blood, you have platelets, little cell fragments, and their job is to go ahead and induce blood clotting whenever you have a cut. So let's say, for instance, maybe you're in your kitchen, you were trying to make yourself a little snack and you weren't paying attention and you kind of cut your finger. Oh, that could be painful. Now, when you see the blood coming out, part of what your body will do is it will go ahead and alert the platelets to come over to the site of injury so that it can form a clot and the blood stops bleeding out, right? The problem is that when you send the stimulus and one platelet shows up, that one platelet is not enough to do the job. So instead, when one platelet shows up, the signal is amplified so that a second platelet will show up. Even more amplified so that a third, a fourth, a fifth, and it just keeps amplifying more and more until you have so many platelets that they can clot together and pop open. And these little green strands that you're seeing on the picture, this is called fibrin. It's a sticky protein that the clots have. That's what they use to kind of seal up the wound. So as you can see, this is an example of a positive feedback system because one platelet wasn't enough for the stimulus to go away, but instead one platelet caused the stimulus to amplify, thereby recruiting additional platelets along the way. Another example I want to give you of positive feedback system is childbirth. So when a woman is about to give birth, what we see happening is that the baby will rotate so that its head will start knocking on the uterus wall. And when it knocks on the uterus, it sends a signal to the brain of the woman to release oxytocin. And this hormone will then travel back to the uterus and cause the muscles to contract. The problem here is that one contraction is not enough for the baby to be expelled out of the body. Instead, that one contraction will cause the brain to release even more oxytocin. And the more oxytocin you release, the stronger and the more numerous the contractions become. So in the beginning of labor, the contractions are relatively far apart, and as labor progresses, the contractions get closer and closer and closer and stronger because your body is flooded with oxytocin, thereby allowing for this amplification signal to occur. Finally, the contraction strength will be strong enough that the cervix will be open enough for the baby to go ahead and move through the birth canal. There's your positive feedback system. And the cool thing is that the oxytocin that's left in the body is then used for emotional bonding because oxytocin is what we often call the cuddle or the love hormone. So by doing skin to skin contact when the baby is put on the mom's chest, it promotes emotional bonding due to the high levels of oxytocin. It works the same for males. So when the baby is placed on the father's chest or on the male's chest, they also release oxytocin and it also helps them emotionally bond with their newborn. And in case you're not interested in childbirth, oxytocin is also released anytime when you look at someone and you have that feeling of infatuation or you like someone. It's the look of love in many of our eyes. It's hormone that we release quite often, especially when we would like to do emotional bonding. Alrighty, I'm going to stop here for part one, and I hope you join me for part two for chapter four. There we're going to take a look at passive and active transport. All right, before I leave, please remember if you have any comments, questions, concerns, please do not hesitate to reach out. All right, everyone. We shall chit chat again soon. Bye.